Let's say this tissue has been connected to electrodes, one side with a positive electrode, another side with a negative. So the influx of these positive ions creates a depolarizing current which is moving in this direction. It is called vector. Now in the ECG paper, if the direction of deflection above the baseline or the isoelectric line, it's called as a positive deflection, and this positive deflection is produced due to the vector which is moving towards the positive electrodes is creating a positive deflection. If the positive ions are moving outside the cell, or efflux of this positive ions creates a repolarizing current which is moving in this direction. Since the vector is moving away from the positive electrode or towards the negative electrode, so the direction of deflection will be below the isoelectric line. It's called a negative deflection. Now let's have some basic discussion on isoelectric line or the baseline. This is represented as a straight line on a ECG paper where there is no positive or negative charges of electricity to create a deflection. This represents the resting membrane potential of the cardiomyocyte, and with some exceptions for this. AV nodal depolarization is an exception where even though the electrical activity or a depolarization is taking place in the AV node, the vector is failed to produce, or the deflection in the ECG paper is nil. It is because AV node is a very tiny tissue, so that the vectors produced by it or the electrical changes happened in the AV node is very less, that the electrode cannot pick up the impulse. Now the direction of deflection depends upon two factors. So the direction of spread of electrical forces or the action potential, and the location of recording electrodes. In simple, an electrical impulse moving towards an electrode creates a positive deflection, while an impulse moving away from the electrode creates a negative deflection. Height of positive deflection and the depth of negative deflection are measured vertically from the isoelectric line. This vertical amplitude of deflection is a measure of its voltage in millimeters. So the magnitude of deflection depends upon the quantum of electrical forces generated by the heart and the extent to which they are transmitted to the recording electrodes on the body surface. If the chest wall is thick, the magnitude of deflection are smaller than the normal, since the fat and muscles intervenes between the myocardium and the recording electrode. So let's say, for example, we have a thick myocardial tissue on one side and thin myocardial tissue on other side. So the vector produced in the thick myocardial tissue is larger than the thin, and the amplitude or the magnitude of deflection is also bigger. Let's say it has some 3 millimeters, and in the thin it is 2 millimeters. Finally, activation of atria and ventricles. So the activation of atria occurs longitudinally by contiguous spread of electrical forces from one myocardiocyte to another. On the other hand, activation of ventricles occurs transversely by spread of electrical forces from endocardial surface to the epicardial surface, that is from inner side to the outer side.